Hey guys, in this second part of the Chapter 3 lecture video, I wanted to go over some of the terms associated with the structures inside of the cell. And so again, we'll refer back to figure 3.1 that just shows you just a generalized picture of a cell. It's a good way to kind of visualize and study some of the main parts as we go through. And so we'll start with the nucleus. And one thing that you should do is to make a flashcard maybe for each of these terms, for each of the vocabulary terms for the cell parts, and that'll help you kind of remember and practice each of the terms associated with the cell. And so first of all, we'll start with the nucleus. This is an example of an organelle. Remember we said an organelle is just a small organ inside of a cell, and usually you're going to find that nucleus somewhere centralized, not always, but somewhere centralized in the cell. Notice it says that the nucleus has a nuclear envelope around it. This is just another name for a membrane. It has a membrane around that nucleus, and that basically has an outer layer and an inner layer, and a little space in between those two layers of that membrane. However, that membrane does have specialized openings or little pores that allows certain materials to pass in and out of the nucleus. And so in this diagram, you kind of see some of those things we just talked about. In section A up here, it's kind of small here. You may need to look at your textbook to see this a little closer. But section A shows you the cell. Has a nucleus somewhere. It's kind of highlighted here for you. You enlarge it, the nucleus is here. And the membrane or the nuclear envelope is shown here on the outside. And then you can also see that it has openings or pores that allow certain things to pass through. Then the other pictures are actually microscope images. Now these are electron microscope images below. This was taken with what's called a transmission electron microscope. They enlarged it 20,000 times, but that allows you to see the actual membrane around the nucleus, and you can even see places where you have little openings or pores throughout on this particular slide image. And then remember that inside of the nucleus is where you're going to find the DNA of the cell. And so in this slide, or this figure 3.14, shows you kind of how the DNA would be found inside the cell. Here's again, that same diagram with the nucleus, and then on the inside of that you would find chromosomes. Chromosomes are where the DNA is tightly wound up into it. Here's your DNA helix over here at the bottom. And that's a ladder, like twisted like ladder sequence of nucleotides, which we'll mention later. The nucleotides are twisted together here, coiled up, and then all of that's coiled around some proteins, and all of the proteins are kind of coiled up into what's called chromatin, and that chromatin is coiled even tighter into what's called a chromosome. Now another organelle that you'll find in the cell is what's called a ribosome, and make sure you make the connection between ribosomes and protein synthesis. Ribosomes are where proteins in the cell are actually made. They're actually produced in ribosomes. Now you'll find ribosomes either one of two types. They'll either be attached to another organelle. For example, there's another organelle we'll talk about called the endoplasmic reticulum. So sometimes you'll find ribosomes attached directly to this membrane system called the endoplasmic reticulum, or you can just find them free, kind of free floating in the cytoplasm. And so they're not attached, but they're basically there to produce proteins. And you can kind of walk through this diagram on your own. I'll show you a few things here. This is again showing you the nucleus here. And so this would be outside of the nucleus, but inside of the cell. And this is showing you an example of a ribosome, and it's going through and producing a type of, getting ready to produce a type of protein here at the ribosome location. We'll go ahead and talk about the endoplasmic reticulum. This is one of a couple different types of membrane systems inside of a cell. And there's, again, two different types here. Remember we said that ribosomes can be attached to the endoplasmic reticulum, and so if ribosomes are attached, then the endoplasmic reticulum is what's referred to as rough. That's because it kind of looks rough. The texture of it looks rough because it has these little small organelles, ribosome organelles attached to it. And so it looks kind of rough, and so that's where it gets its name from. And so the rough endoplasmic reticulum, because it has ribosomes attached, mainly functions to produce proteins. If there are no ribosomes attached to the endoplasmic reticulum, then it's referred to as smooth. And so if it doesn't have ribosomes, then it doesn't make proteins, and so it does other things. It can generate lipids, and so it can go through fat production or lipid synthesis. It can help with cellular detoxification, getting rid of some harmful chemicals in the cell, and it can actually store some calcium in a specialized type of endoplasmic reticulum that you'll find in muscle cells, and that'll be covered in a later chapter. And so in this picture, you kind of see an outline, again, of the cell here, the nucleus, and then usually surrounding the nucleus, you'll find the endoplasmic reticulum. And so in the large portion of the diagram, up at the top portion corner would be inside the nucleus. Notice that the membrane of the nucleus, or the nuclear envelope, is continuous with the membrane system called the endoplasmic reticulum. The first little bit of this is called rough because it has these little small circles. Those circles are ribosomes. And so the first part of this is mainly there to produce proteins. And you have a section of endoplasmic reticulum after that known as smooth, which is going to accomplish the other functions. Now, another membrane type system that you have in your cells is what's called Golgi Apparatus. Sometimes it's called Golgi Complex. I've seen it called the Golgi Body. So you have different names for this, but this is basically where different types of molecules are going to be packaged up into vesicles so that they can either be stored or secreted from the cells. And so it can collect, modify, package, and even distribute different types of molecules, including the proteins that were made by the ER, the lipids that are made by the ER. They can go into little storage compartments. Those storage compartments are known as vesicles. And then those vesicles can store up these molecules, and they can also eventually release the molecules in the process called, remember we talked about exocytosis earlier, and so it can release those molecules outside of the cell. And so in this figure 3.13 shows you just kind of what a Golgi apparatus would look like. There are no set locations for this. This is just inside of the cell, inside of the cytoplasm. I think usually you're going to find it close to the border of the cell. That's because it's going to secrete or package up chemicals and molecules inside of these vesicles, which can be stored in the cell and eventually fuse with the outer membrane of the cell in that process called exocytosis and releases molecules out of the cell. Now, a type of storage vesicle is called a lysosome. This is one example of a vesicle that's formed by that Golgi apparatus. And lysosomes generally contain enzymes. And so enzymes are specific types of molecules that help to speed up chemical reactions, and they also help to digest things. And so inside the cell you have little storage vesicles called lysosomes that will store up some digestive enzymes for the cell. Now, one example of why you would need these digestive enzymes is to break down any type of cellular debris or maybe a foreign cell that has been engulfed. So for example, white blood cells, one of their main jobs, some of them, is to go around and get rid of bacteria. And so when the white blood cell, it can phagocytize, it can go around and engulf or eat the bacterial cell, incorporates that bacterial cell inside of the white blood cell, and then the lysosomes release enzymes that will come around and actually digest the bacteria and get rid of it. And so lysosomes, again, they're a storage vesicle, they're formed from the Golgi, and they contain enzymes. And figure 3.18 will show you kind of an example of that. Here you have some kind of cell. Maybe this is a white blood cell. It has phagocytized something, as taking something into the cell, and now it's stored something. It could be bacterial cells or some type of debris. Something inside now is compartmentalized inside this vesicle. And here's your Golgi again. The Golgi has made a lysosome with enzymes in it. These two fused, come together, and those enzymes are released into that vesicle to help break down whatever that vesicle was storing. Now, another example of a vesicle, and like I said, a good way to study all of these is to just make a flashcard for each one. Make a flashcard for the nucleus, the Golgi, endoplasmic reticulum, ribosomes. Just go down the list, make some flashcards, study vocabulary, look at the diagrams. That'll help you as well. But this is another example of a membrane-bound vesicle, also produced by the Golgi, called a peroxisome. The name here tells you what's inside of it. Peroxisome looks a lot like peroxide, and so this stores enzymes that help to break down peroxide. Hydrogen peroxide sometimes is produced by the body by certain metabolic pathways in the body, and it's toxic to a cell. You got to get rid of it. And so the peroxisomes are vesicles that have enzymes, specialized enzymes that help to break down different molecules, including fatty acids, some amino acids, and even hydrogen peroxide can be broken down. Now we're moving on into the mitochondria. Notice this spelling up here, mitochondria is plural. If you see the word mitochondrion, it's just referring to one, mitochondrion. That's one or singular version of this organelle. And so make sure you make the connection here between the mitochondria and energy production. This is where the majority of ATP is produced. There'll be another chapter later on where we go through and talk a little bit about cellular respiration or aerobic metabolism, and that's taking place mostly there in the mitochondria. And so this is also, again, an organelle that has membranes. It actually has two main membranes, an inner and an outer membrane. And those are the locations for ATP production. And so in this diagram 3.19, you see an example of a mitochondria drawing over here. It does have the outer membrane on the outside, it has an inner membrane on the inside, and then it has this inner membrane that kind of curves around all throughout the inside. That's called a crista membrane. And you have, this is where a lot of the biochemical reactions take place to produce the ATP energy. And in this microscope image, again, this is an electron image showing you what this would look like if you enlarge this, magnified this 34,000 times. Now, your cells have a type of framework inside of it known as the cytoskeleton. It's not exactly a skeleton like you think of as a skeleton, but it does provide framework inside. And there's some examples here. It basically consists of protein molecules, and it does support the cell, helps to hold those organelles that we've just been talking about in place. Remember, there's cytoplasm in the cell, which is like a jelly-like substance, but you have the cytoskeleton that helps to hold everything in place. And that also maintains kind of the shape of the cell. Now there's different examples, different structures for the cytoskeleton, and that's listed here at the bottom: microtubules, microfilaments, and intermediate filaments. So first of all, microtubules, this is just one example of the cytoskeleton network that you have. Microtubules are hollow, and again, they're formed from proteins. You'll find microtubules in a couple different locations within a cell. One of the area is it helps to form certain structures that play a major role in cell division. And so one of the parts of cell division is to move chromosomes around in the cell. And so the microtubules form what are called spindle fibers that helps to move different chromosomes around in the cell. It also helps in forming what are called cilia and flagella. Both of these structures here are organelles that are both for movement, some kind of locomotion or movement. microfilaments are a little smaller, and that's why they use the word micro in front. Microfilaments are filament-based. They're still protein, and they mainly are there for structural support. They do seem to have some play some role in moving the cell around, or at least changing the shape a little bit. They allow for muscle cells. They're a little bit differently shaped than most cells. They allow for muscle cells to kind of stretch out and contract a little bit. And so microfilaments play a role in muscle contraction. Then you have the last type of protein filament, known as an intermediate filament. And this is, as the name implies, this is an intermediate size. It's smaller than the microtubules, but a little larger than the microfilament. So that's where it gets its names. It's an intermediate filament, or fibril, which just means protein filament. And again, they're basically there for supporting the cell. Sometimes you find, you may have heard of the protein known as keratin. That's associated with skin, with your skin cells. And so some of your skin cells have this protein there called keratin, which is basically an example of an intermediate filament. So in this picture, figure 3.20 shows you, again, that example of the cell that we've been looking at, and it shows you inside the cell, and it shows you some examples of how the cytoskeleton would be located and kind of where it's located. Again, you have some of the microfilaments that are inside. You have microtubules, and again, those are going to be serving different functions. And then the intermediate filaments, a lot of these are kind of there for structural support. It kind of makes like a lattice or a kind of a scaffolding system inside of the cell there for support. Now, I mentioned the microtubules help to form something called the spindle fibers, and they also make something called a centriole. The centrosome is an area located in the cytoplasm. It's usually close to the nucleus where the microtubules are actually formed. And that centrosome region has what are called two centrioles. These are another example of an organelle. And usually you'll see these kind of set in a cell end to end or perpendicular to each other. And this is a location of where microtubules will be produced, and that helps in the process of one type of cell division, which we'll look at shortly, called mitosis. And so this is kind of where the what this is going to look like. Again, here's our picture of the cell. In the area kind of offset there from the nucleus, you would have the centrosome, that location inside the cytoplasm, where you find the centriole, and kind of see the centriole is here, and it kind of forms each one of these tubes would be the microtubule. There's a certain arrangement pattern that you see here with three. It's a 9-3 pattern. You have 3 tubules, and then you have 9 places going around. Now, another example where you find the microtubules would be cilia. Again, cilia and flagella are both for movement, some type of locomotion or movement. And so a lot of times you find cells, or sometimes you'll find cells that have cilia on the kind of the surface of the cell, and that cilia moves, can kind of sweep things up. For example, in the respiratory tract, you have cells that are lined with cilia, and that kind of sweeps up particles and mucus and things out from the respiratory tubes, so that you can kind of get all of that mucus up out, so it doesn't settle down in the lungs. That's one example of cilia that you would find in the body. Flagella, again, also made from the centrioles. Flagella is similar to cilia, but usually they're larger and longer, and you generally just find one flagellum. That's singular, so if you see this word, that's singular. You usually find one flagellum per cell, versus cilia, which are smaller, and you would find many cilia per cell. And so one example where you would find flagella would be the male gamete cells, the sperm cells. They would have a flagellum that allows for the movement of the sperm cell. Now, one other area that you would find some microfilaments or cytoskeletons, what's called microvilli. And so on the cell membrane, many times you find these extensions coming out of the cell membrane. They're not really there for movement like cilia and flagella, but many times they help to increase the surface area of a cell. So anytime you see microvilli, many times make the connection of increasing the surface area. And so places where you would generally find microvilli are places where you need more surface area. Rather than having a smooth surface going across, you would have kind of a surface with many little projections, very small projections coming out. Places that you need a lot of surface area would be the intestines, because we'll get to this in a different chapter, but many of you realize the intestines help to absorb a lot of nutrients. Well, the more surface area that you have, the more nutrients you can absorb, and that is helped by having that microvilli. Kidneys, same type of thing. The kidneys are there to reabsorb extra fluid, extra water. It allows you to excrete the waste, but also reabsorb a lot of water and fluid back into the bloodstream. And so again, you need to be able to absorb lots of fluid, and so you would need microvilli for that. Now, the last couple things talks about your genetic material. We're going to go through a few of these things. Be sure to kind of read over some of this chapter. I'm going to just give you a quick outline of some of these topics here, and then we'll talk about, lastly, we'll talk about the cell division process known as mitosis. First of all, make sure you understand what DNA is. This is a type of nucleic acid that is located inside of your cells, mainly found in your nucleus, but you do have DNA in your mitochondria as well. And so you have a specialized type of DNA there called mitochondrial DNA, but the majority of your DNA resides inside of the nucleus. And this is going to give the directions or the genetic code so that you can create all the different proteins that you have in the body. And that process is known as gene expression. And a little bit about the structure. The DNA is made out of, like I said, it's a nucleic acid, but it's made out of subunits called nucleotides. And you have basically a string or a strand of two nucleotide strands, and they come together, they kind of twist together, and that creates what's called the double helix. And so each strand of the nucleotide is going to have three parts to it. It has a 5-carbon sugar, it has a phosphate group, and it has a nitrogenous base, or a nitrogen-containing base. And so you have each nucleotide is going to have these three parts to it, and then you'll have a chain of those hooked together to create the DNA strand. And so each of the nucleotides creates a particular pattern, and that creates a gene sequence. And the order of those nucleotides, there's 4 nitrogen bases, adenine, guanine, cytosine, and thymine. The order of those 4 nitrogen bases will determine the sequence that will determine that gene. And that gives a particular set of instructions to make a particular type of protein. Gene expression, which just briefly mentioned a few things, basically describes protein synthesis. It involves turning on a particular gene and then taking that genetic information from the DNA and transcribing it into another type of nucleic acid known as RNA. And so transcription is the process, again, where you take and you copy the DNA code into what's called messenger RNA. That messenger RNA, this process is taking place in the nucleus. The messenger RNA will leave the nucleus. It'll go through those nuclear pores, like we mentioned, go into the cytoplasm, go to the ribosome. That's where protein synthesis is going to take place. That messenger RNA basically carries the message of the DNA in the RNA form over to the ribosomes and starts to build the protein. And so translation, that's known as translation, that's just another name for protein synthesis. So protein synthesis and translation are saying the same thing. You're taking messenger RNA, you're translating it into a protein. It starts out as a nucleic acid, but you're translating it into a protein. Just a couple things about transcription. Remember, I've already mentioned this, but transcription takes place inside the nucleus. Transcription is using the DNA as the genetic code to make the structure of messenger RNA. And during the process of transcription, that double helix of the DNA has to separate. There's certain enzymes that come in and basically unzip or unwind that DNA strand. And then you start to build an RNA strand that is complementary to those DNA nucleotides. And that will create your messenger RNA. There is some more information in the chapter, and it'd be good to kind of look over, use the diagram here, kind of go through the steps, and you can kind of see the outline here. Here's your nucleus again. This is where you're going to find the DNA. This is where your chromosomes would be. The DNA is going to unwind itself. It's going to go through the process of transcription inside the nucleus, create the messenger RNA molecule that comes out. There's those pores again, comes out through the pores, goes into the cytoplasm. Here's a much enlarged version of a ribosome, and this is where protein synthesis or translation is going to take place. Now, the last topic I wanted to go over for this chapter is the cell cycle, and so we'll talk about this. Cell division is the process of replication or replicating a cell, and that cell cycle really has two major phases. One is called interphase, and that's during a time where the cell is kind of growing and developing and preparing to reproduce itself. Then you have the dividing phase, which is called mitosis. That's where you've got several steps that's going to take place that actually goes through and takes a cell and divides it in half and creates two identical cells. And so some things that are good to know about cell division or about the cells. Each human cell basically has a total of 46 chromosomes. You have 23 pairs of chromosomes. Now that's a little different from your gametes. We'll talk about this in a different chapter, but your gametes, your egg cell, and your sperm cell are haploid. They have half the number of chromosomes. So these cells, the egg cell and the sperm cell, will only have a total of 23 chromosomes. Whereas your other cells, so for example, a skin cell or a liver cell or a muscle cell, would have a total of 46, 23 pairs. You have 23 from each parent inside of that cell, and that gives you a total complement of 46. That's what's called a diploid cell. You have two copies of each chromosome. Now, one pair of those chromosomes included in that 23 count are sex chromosomes, and if you have two X chromosomes as your sex chromosomes, then the person is a female. It's the Y chromosome that determines if the person is male, and so if you have X and a Y, then you'll have a male individual. Now, there are two types of cell division. We're going to focus on mitosis here, and we'll talk a little bit more about meiosis in another chapter, but mitosis is the process where you basically take a single cell, a parent cell, and it divides into two daughter cells. That division should give you two identical cells at the end of mitosis. Now, there's four main phases to mitosis, beginning with prophase, metaphase, anaphase, and then telophase at the end. And so what I'll do is just quickly give you a few bullet points about each of these stages. Prophase, this is your first one. This is where you first are able to visualize chromosomes. Remember the chromosomes that are in the cell. Usually they're in long, thin strands called chromatin.